Good. Yeah, so the T cell therapy, as was just described, I think can be quite effective, but it can also be able to target tumor cells or normal cells that have limited numbers of target antigens. And so one has to think about this. I think the CD19 is an excellent target because of its specificity on B cells, but as was mentioned, when you do target CD19, you do deplete B cells, resulting in profound hypogammaglobulinemia and the need for sustained support with intravenous immunoglobulin. So the idea of trying to find surface antigens that might be more specific to the target population and less distributed on normal cells is actually appealing. And ROR1 is actually an interesting antigen that came about not only through microarray studies, but later when the patients were immunized with cellular vaccines, as you recall. We were doing a lot of work with this with CD40 ligand transduced leukemic cells. We noted that some patients parenthetically make anti-ROR1 antibodies. And these are antibodies, some patients actually had antibodies which were quite active against the target protein, and parenthetically they did quite well clinically. We were interested in trying to pursue this further. I know that Dr. Bird's group also observed the development of anti-ROR1 autoantibodies in some patients treated with lenalidomide. I'm not sure what the clinical course of those patients were, but it's interesting that you can develop autoantibodies against this and not fall apart at the seams, so that the development of anti-ROR1 antibodies internally can be tolerated. ROR1 is an embryonic antigen, not a fetal antigen, but it's expressed in early development. This is the expression in early developing mouse embryos. It's a type 1 tyrosine kinase-like protein, and it has a number of interesting domains, and our laboratory is spending a lot of time trying to understand what the biology of this is, and that's, suffice it to say, it's a whole nother talk. But what's interesting is that ROR1 is expressed in embryonic development, you see, and then attenuates even during fetal development, such that at term the expression level is quite limited. And more importantly, the expression of ROR1 is not present on certain stem cell populations, such as hematopoietic stem cells. We have done tissue search. Actually, when you develop some high affinity, very specific anti-ROR1 antibodies, the specificity does appear quite limited to CLL and other types of tumors. But the one exception is the expression of this on hematogones, which are these strange types of precursor B cells which have CD10, CD5, and CD19 and low levels of CD45. These are more commonly seen in patients recovering from myeloablative transplant, particularly pediatric patients, but are present in very minute quantities in normal marrow and may not represent an obligate step in B cell development. This is work that I don't have time to present. Now, the expression of ROR1 is independent of the expression of some of the prognostic markers, such as ZAP70 or mutation status, and it's not expressed on CD5 B cells of normal adults, as you see in histogram there. So normal B cells are lacking this antigen. And the expression level is quite uniform on a number of CLL patients, and we've actually looked at more with greater than 94% of the patients expressing the ROR1 surface antigen, and some data that we have that may indicate that ROR1 is a property with CLL, and the absence of ROR1 may define a different entity. But if we look at some of the antibodies developed, we've generated a series of antibodies and tried to map out the epitopes, but also define the specificity of the binding to normal tissues, and both in our lab and we outsourced this to a comparative biosciences. And you could see lack of cross-reactivity of a number of tissue organs, including pancreas and adipose tissue. There is some expression in various cancers. This is infiltrative pancreatic cancer here that's expressed in the ROR1. And we've generated single-chain antibodies that seem to have similar affinity. Actually, this is a Connexa measurement, and the affinities that we're striving for in the nanomolar or subnanomolar KD. And so we have generated a number of these, but this is one in particular. This is Jay's review that he presented earlier. There's an evolution of the types of chimeric receptors. I think second generation is referred here, and third generation. The differences being the internal cytoplasmic domains, be it the accessory molecules of CD28 or CD137. I think that this is going to be an evolving process to define not only what types of chimeric receptors are the best, what type of T cells are best to have these chimeric receptors expressed, and how to activate the T cells. So this work is going to be ongoing. I think hopefully it'll improve upon the response rates that were discussed earlier. Now, in contrast to the Memorial Sloan Kettering and also the approach that was used at Penn, both use retroviruses in case of the Penn using lentivirus vectors. Lawrence Cooper has adapted a strategy to use electroporation. And what you do is you have a plasmid which has a transposon element that can allow for integration, provided that you have a transposase specific for that transposon element. In this case, the transposase comes from salmon. And so you co-transfect in with the salmon transposase with the transposon plasmid, and then you can get integration fairly with high efficiency after electroporation. These cells, however, do require to be expanded on antigen-presenting cells, which can be engineered to express the target antigen, plus a number of other key features, including the 4-1BB ligand, also membrane IL-15, and in the presence of IL-2 and IL-21, and also the expression of CD86. And there's also an Fc receptor, so you can dial in other types of surface molecules to expand specific types of T cells, be it type of naive versus memory. And this is something that's work ongoing that Lawrence has invested a lot of time and effort in to see how we can get specific types of T cells to expand. But these then are expanded, and then later on they're infused into the patient, similar to the lentivirus vectors. Now this process takes a little bit longer, but it's actually been minimized to maybe a 10-day period, so that's reasonable. Maybe it could be done shorter, but this is actually appealing because you're not using a retrovirus construct. The expansion of different types of constructs that have been generated, either first, second, or third generation, seems to be very reasonable and similar to the CD19 CARs that he generated through the similar technology. And also the specificity appears to be quite high. These are target cells that express the ROR1 and the CD19. And you can see that in the red here is the ROR1 specific CAR, and these are the CD19 specific CAR. So this cytolysis seems to be quite good. This is a ROR1 specific CD19 specific target. You can see the CD19 CARs going after these, but the ROR1 not, and vice versa. Here, this is a ROR1 specific tumor target, and you can see that the deletion is here with the ROR1 CAR-bearing T cells, but not with the CD19 bearing CAR T cells. And in vivo, this appears to be quite effective, and this is work that's ongoing too to see this is without treatment a ROR1 positive type leukemia transplanted into these immune deficient mice, allows for rapid engraftment with the second and third generation CARs. You can see that you can get protection of these. They haven't required any anti-IL-6 antibody, but perhaps that might come about if this was administered to patients. So we're very excited about the prospect of getting this into the clinic, and certainly we hope to do that in the near future at both MD Anderson and UCSD.